Okay, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, so what we've now discussed is the process of nociception, which is the process by which noxious stimuli can be converted into receptor potentials, and we've also now discussed how that receptor potential is going to contribute to the formation of an action potential, although we don't at present know where the site in the nociceptor is where action potentials are first generated. Okay, that action potential is now going to propagate up the peripheral process of the primary nociceptive afferent, and it's then going to propagate on to the central nociceptive process of that primary nociceptive afferent, and that central process is going to go and synapse onto a neuron that's in the spinal cord. So it's this that we now want to discuss. Okay, so first thing then that I'll just redraw out is this picture of our primary nociceptive afferent. Okay, so here is the cell body with this single process coming off the cell body, which then splits into the central process here, okay, and then the peripheral process going the other way. Right, so what we now want to see is where the cell body of these primary nociceptive afferents is, and we also want to see where is the central process going to go, and what neurons is it going to synapse onto, and what then do they do next. Okay, right, so the first thing I'm going to do then is I'm going to talk a little bit about the anatomy of the spinal cord. So what we're going to do is we're going to imagine that we have cut the spinal cord and we're looking at a transverse section of it. So we're going to cut it in a transverse plane and look at a transverse section of it. Okay, and this is what you end up seeing. Okay, so it's in a sort of oval shape, like so. Okay, and then at the front what you're going to have is a fissure inwards like so. Okay, and at the back you'll have a septum coming into the spinal cord. So here is the spinal cord outline drawn here. Okay, and then you'll have a septum coming into the back like so. So let me label up these structures at first. So this is the ventral fissure of the spinal cord that runs all the way along the length of the spinal cord. So on the anterior surface of the spinal cord there is a little fissure known as the ventral fissure. Okay, and at the back there is a little invagination of the cortex of the spinal cord, the outer surface, inwards, and this is known as the dorsal septum of the spinal cord. Okay, right. The next broad anatomical features that I want to highlight of a transverse section of the spinal cord are the division of it into white matter and grey matter. Okay, so I'm going to draw the grey matter portion out now. Okay, and it's in this sort of strange, sort of butterfly-like shape here. Okay, with these two horns, as it's called, that are dorsal horns, and then you also have these two horns that are ventral horns here. Okay, like so. So that's the grey matter. This great big section here is the grey matter of the spinal cord, and all the bits outside of that are the white matter. Now, what's the difference between grey matter and white matter? Well, basically, grey matter is where the cell bodies of neurons are, okay, and white matter is where the axons, which are myelinated, are, and that's why it appears white, because the myelin appears white. Okay, right. Now let me label up the different columns of white matter. Or in fact, firstly I'll tell you about the horns of the grey matter. Okay, so you can see that the grey matter of the spinal cord has these sort of horns extending off. Okay, so these two horns that are at the back, this one here and this one here, these are what are known as the dorsal horns. Okay, and of course you have two dorsal horns. You have a left dorsal horn, which is this one here, okay, and you have a right dorsal horn, which is this one here. Okay, you might think, but that one's on the left-hand side, but think about it from the patient's perspective. Okay, this is the front and this is the back. This is the patient's left and this is the patient's right. So this is the left dorsal horn, this is the right dorsal horn. Okay, I won't put an arrow to the other one, even though I should, because I have put dorsal horns rather than horn, because I need the other one for showing the laminae in a moment. Okay, so I'm going to put all the labels on this left-hand side and save the other side for showing the rexed laminae of the grey matter in a moment. Okay, right, so that's the dorsal horn. Okay, and then we also have these horns towards the front, and these are known as the ventral grey matter horns, or just the ventral horns of the spinal cord. And again, we have a left ventral horn and a right ventral horn. Okay, so that's the division of the grey matter up. Okay, the other thing that I'd like to add on to this picture is that there is a canal down the middle, okay, which is known as the central canal of the spinal cord, and that's full of cerebrospinal fluid. Okay, and I think I'll colour this in to have a little bit more colour on here. So that's the central canal there. Okay, right, now what I want to do is split up the white matter columns, okay, and tell you about what their names are. Okay, so you can see that there are these two columns right at the back here, a left one here and a right one here. These are known as the dorsal columns, okay, and I might colour them in turquoise. So this is a dorsal column. That's the left dorsal column specifically that I've coloured in, but you have the same thing on the right hand side. So that's the dorsal column, okay, which I'll underline in turquoise. Okay, right. Then another white matter column, which I'll highlight in orange here, is this one here. Okay, that's known as the lateral column, the lateral white matter column of the spinal cord. And again, you have two lateral columns, a left one, which is the one we're showing here, and then the right one's over here. Okay, so that's the lateral white matter column. Okay, then you have the anterior white matter column, which is in green here. Okay, so this is what's known as the anterior white matter column, or just the anterior column. And again, I've highlighted the left anterior column, but there is also a right anterior column. Okay, and then that space in between the anterior column and the lateral column here, in vivid purple, that's what's known as the anterolateral column. Okay, so that's called the anterolateral white matter column, and we're going to need this anterolateral white matter column. The secondary neurons in our pathway are going to ascend in the anterolateral white matter column, so we'll be seeing a lot more of this column in a moment. Okay, right, so that's some basic anatomy of a transection of the spinal cord. Okay, what we want to look at now is the rexed laminae of the grey matter of the spinal cord is divided into. But before we just do that, I also would like to show you a dorsal root and a ventral root coming into the spinal cord. Okay, so let's imagine that we've taken this transverse section of the spinal cord at a specifically good level, so that we've cut it in the middle of a section where there is a dorsal root coming out the back and a ventral root coming out the front. Okay, so let me just show this here, and I'll show this now for this right hand side and not for the left hand side, but it would also be equivalently on the left hand side. Okay, so this is the dorsal root here. This is the dorsal root ganglion that I'm showing as this swelling here. Okay, like so. And here is the ventral root here, like so. And then they combine together to form the mixed spinal nerve. So let's put some labels on here. So basically, out of the spinal cord emerges two roots, and this is at multiple different levels. Okay, this root here is what's known as the ventral root. Okay, and the back root at the back is then known as the dorsal root. Okay, and the two then combine together as the mixed spinal nerve here. So this is a mixed spinal nerve, and it will be at a specific level, so there's loads of mixed spinal nerves. Okay, so for instance, you have all of the C1s, then all of the T1s, then all of the the lumbar ones before that, and then the sacral ones. Okay, so there are loads of spinal roots. Okay, right, so we've taken it at some specific level such that we have got a ventral root and a dorsal root shown here. And of course you'd have the equivalent on the other side. Now, the ventral root contains axons of neurons which have their cell body in the ventral horn and which are coming out of the spinal cord, and these will include things like motor neurons which innervate skeletal muscle cells. Okay, the dorsal root contains the axons of neurons that are coming into the spinal cord, and this is where the sensory neurons come. Okay, so our peripheral process of our primary nociceptive afferents is going to come in through our mixed spinal nerve here. It's going to come in to the dorsal root, and then here, finally, this is where the cell body of that primary nociceptive afferent is actually going to be. So this, first I'll highlight up the peripheral process. Here's the peripheral process. Then we finally got to see where the cell body of our primary nociceptive afferent actually is. And then the central process is going to continue on in the dorsal root and actually go into the spinal cord, and it's going to synapse on neurons that are in the dorsal horn of the spinal cord, basically. Okay, so here, this is the central process going on here. Okay, right. So now what we want to see is where are these primary nociceptive afferents actually going to synapse. Okay, and for this we need to discuss the rexed laminae of the grey matter. So basically the grey matter is divided up far more carefully than what I've shown you so far. All I've shown you so far is the dorsal horn and the ventral horn. In fact, it's divided into 10 separate laminae on both sides, known as the rexed laminae. Okay, so this is what we're now going to study. Okay, and we're going to show it for the right-hand side here. So I'll put a division there, splitting the left-hand side from the right-hand side. Okay, right, so there are 10 rexed laminae. Okay, the first 6 are all in the dorsal horn. So I'll show these first. 1, 2, 3, 4, 5, 6. So those are the first 6 rexed laminae. So I'll colour them in separate colours. So here's 1 in red. Okay, here's number 2, rexed laminae number 2 in blue. Here is rexed laminae number 3 in green. Rexed laminae number 4 in turquoise. Rexed laminae number 5 in orange. And then finally rexed laminae number 6 in magenta there. So that's number 1 to number 6, and it's done in Roman numerals. Okay, so 1 to 6, and it just goes in a nice, easy to understand order from outside to inside, basically. Okay, now, it is easier to show rexed laminae 8, 9, and 10 before showing rexed laminae 7. Okay, so that's what I'm going to do. Rexed laminae 8 is right at the front here, and then a part of this portion is rexed laminae 9. Okay, so this here, this is rexed laminae 8 here, okay, and this one is rexed laminae 9, and I was about to write 11. My Roman numerals aren't brilliant. Okay, so here is rexed laminae number 8 in blue there, okay, and here is rexed laminae number 9 in turquoise. Now let's show rexed laminae number 10. That's this portion here, okay, so I'll put that in green. So all of that is rexed laminae number 10, and then all of what's left is then rexed laminae number 7. So this is 10, and then all of this here is 7, which I might try colouring in yellow. Okay, so here is rexed laminae number 7. I don't want to smudge the 7. Yes, got away with it. Or, yeah. Okay, there we go. So those are the different rexed laminae of the spinal cord. Now the ones that are going to be particularly of interest to us are rexed laminae number 1, rexed laminae number 2, and rexed laminae number 5. Okay, so we're actually not going to need all of these fancy ones further along down here. We're actually only going to need these really simple ones down here. Okay, right, so what I'm now going to do is draw a zoomed in picture, basically, okay, of the dorsal horn of the spinal cord. Okay, so here is this dorsal horn. It's separated into these 6 rexed laminae, but we don't actually need rexed laminae number 6. We only need 1 to 5 here, so that's what I'm doing. In fact, the only ones we're going to need is number 1, number 2, and then number 5. So these are going to be the important ones. Okay, so basically the secondary neurons, which are the neurons that the primary nociceptive afferents are going to synapse on, are going to be in rexidlamine number 1 and rexidlamine number 5. Okay, and these secondary neurons are going to be called spinothalamic projection neurons. Okay, because they are going to project up to the thalamus from the spinal cord. So spinothalamic projection neurons, or you can call them the secondary neurons if you like, the secondary nociceptive neurons, whatever you want to call them. Okay, but I will probably refer to them as the spinothalamic projection neurons. Okay, so the spinothalamic projection neurons are going to be in rexidlamine number 1 and number 5. Okay, so here are their cell bodies. Here's one, and here is another one in rexidlamine number 5. Okay, so we now want to look at how the primary nociceptive afferents are actually going to synapse onto these spinothalamic projection neurons that have their cell bodies in rexidlamine number 1 and rexidlamine number 5. Okay, so this is where it differs depending on whether you are a delta fiber primary nociceptive afferent or whether you are a C fiber primary nociceptive afferent. Okay, so let me explain this. So basically, for the ones in rexidlamine number 1, it's exactly the same for A delta fibers and for C fibers. Okay, so all of them will come in like so, and they will synapse onto dendrites. So I'll have a dendrite shown here. Okay, so that little sticky bit out there is a dendrite. Okay, and again, here is another central process. So we'll say that this is one from a C fiber primary nociceptive afferent, and this one here is from an A delta fiber primary nociceptive afferent. Okay, so these are central processes here, and they'll both come into rexidlamine number 1 and synapse on the dendrites of these spinothalamic projection neurons in rexidlamine number 1. Okay, now coloured in orange. Okay, but it's more complicated for the neurons that have their cell bodies in rexidlamine number 5. Okay, basically the A delta ones are going to brave it all the way down here to synapse onto dendrites of the spinothalamic projection neurons in rexidlamine number 5. Okay, so if you're an A delta type primary nociceptive afferent, your axon could go all the way into rexidlamine number 5 and then synapse on a dendrite of one of these spinothalamic projection neurons in rexidlamine number 5. But the C fibers, they don't penetrate that far. So instead what actually happens is these spinothalamic projection neurons in rexidlamine number 5 have very large dendrites which go all the way into rexidlamine number 2. Okay, and basically the C fibers can synapse onto these dendrites that are in rexidlamine number 2. Okay, so this here, this is a C fibers central process here, synapsing onto one of these dendrites that has ventured all the way up into rexidlamine number 2. Okay, now there is a special name for rexidlamine number 2 that people will also use, and I've missed a word out. It's called substantia gelatinosa, and I was just about to write the gelatinosa. Okay, so there we go. Substantia gelatinosa means rexidlamine number 2. Okay, so the cell bodies of these spinothalamic projection neurons are not going to be in rexidlamine number 2. However, you can still have these synapses between spinothalamic projection neurons and C fiber type primary nociceptive afferents in substantia gelatinosa or rexidlamine number 2 because of the fact that these spinothalamic projection neurons with their cell bodies in rexidlamine number 5 can send dendrites back into rexidlamine number 2. Okay, right. So let's now talk about what these synapses actually are using. Okay, so these are these central processes of these primary nociceptive afferents coming into the spinal cord and synapsing on these spinothalamic projection neurons. But what neurotransmitters are they actually using here? Well, the main one that they are using is glutamate. Okay, So usually the neurons are releasing glutamate onto the dendrites of the postsynaptic neurons. Okay, and the receptors for glutamate on the dendritic spines of the dendrites on the spinothalamic projection neurons are going to be AMPA-type glutamate receptors and NMDA-type glutamate receptors. Okay, so let me explain what I'm drawing here. This is the axon terminal of one of my primary nociceptive afferents, okay, that has come into the dorsal horn of the spinal cord. Okay, so I might even colour this in blue. Okay, and I should just say that the neurotransmitter released is the same for A delta and C fibers, so that's one complexity that we don't need to worry about. Okay, and this is a dendritic spine coming off one of the dendrites of the spinothalamic projection neurons. Okay, so the axon terminals don't synapse directly onto the dendrite, they synapse onto these little processes coming off the side of the dendrite called dendritic spines. So the dendrites will be covered in these little dendritic spines, and basically it's these that form the interfaces with the primary nociceptive afferent axon terminals. Okay, so basically these will be releasing glutamate as one of their major neurotransmitters, and the dendritic spines will have receptors for glutamate, which will be ligand-gated ion channels, and there will be two major types of ligand-gated ion channels for glutamate used. Okay, one is the AMPA-type glutamate receptor, so we'll say that this one is this one here, so this is an AMPA-type glutamate receptor. Okay, named after one of the exogenous agonists which can open the AMPA-type glutamate receptor, which is called AMPA. Okay, so we'll colour that in red here. Okay, AMPA-type glutamate receptor. And then you'll also have NMDA type glutamate receptors on these dendritic spines as well. Okay, and this is going to become important later on because NMDA receptors are actually incredibly important elsewhere in the central nervous system in long-term potentiation, which has a very important part to play in memory. Okay, and in fact, a similar thing to long-term potentiation, in fact the exact same thing really to long-term potentiation, can occur at these synapses here, and that's plays a big role in central sensitization, which we'll come across later on. Okay, so these are NMDA type glutamate receptors. Okay, right. So both of them bind glutamate and open, although the opening kinetics is a little bit more complicated for NMDA type glutamate receptors. In fact, NMDA type glutamate receptors to open, they not only need glutamate, but they also need the electrical potential difference across the cell membrane to be depolarized. Okay, and we'll talk more about that later on when we come on to the central sensitization. Okay, right. So, glutamate is one of the major neurotransmitters used here. In fact, it is the major neurotransmitter used here. However, it's not the only neurotransmitter that these axon terminals of these primary nociceptive afferents can release. Okay, in fact, when you look at an electron micrograph of one of these axon terminals, you see two types of synaptic vesicles in here. Okay, you see small electron-lucent ones, and then large, very electron-dense ones. Okay, so this one here, this is a small electron-lucent vesicle, okay, which means that on the electron micrograph it looks very clear. Okay, so electron-lucent, that's what that means. Okay, so this is a small electron-lucent vesicle, and these are the vesicles which contain the neurotransmitter glutamate. Okay, but there are also these much bigger vesicles which are very electron dense and therefore appear very dark. So this is a large electron dense vesicle. Okay, and these large electron dense vesicles actually release another neurotransmitter, a different neurotransmitter that's actually a peptide neurotransmitter. Okay, so the other key neurotransmitter that these axon terminals release is, where should I write it? I'll put it up here. Okay, substance P, okay, which is a peptide, it's a polypeptide. Okay, now substance P can act on receptors that are also on the dendritic spines of these spinothalamic projection neurons. Okay, but these are not ligand-gated ion channels. Instead, they are G protein coupled receptors. So they have the characteristic 7 membrane spanning alpha helices that I've just tried to show there. Okay, and the receptor for substance P is what's known as neurokinin 1 receptor. Okay, so the NK1 receptor. Okay, right, so I'll colour that in. So this is the neurokinin 1 receptor, or the NK1 receptor. Okay, so that stands for neurokinin 1 receptor, or just NK1. And basically this is also capable of activating the spinothalamic projection neurons. So just like the AMPA and the NMDA type glutamate receptors, which produce depolarization of the electrical potential difference across the cell membrane when they are opened by allowing ions to move across, when you actually activate the neurokinin 1 receptor through a cascade which will no doubt affect other ion channels on the cell membrane, it leads to depolarization of the electrical potential difference across the cell membrane indirectly, basically. So although this isn't the thing that actually directly produces the change in electrical potential difference, it activates a cascade that then affects some other target, which then changes the electrical potential difference across the cell membrane and in fact depolarizes it. Okay, so both of these neurotransmitters can depolarize the electrical potential difference across the cell membrane, and this will contribute to the spinothalamic projection neuron firing action potentials. Okay, right. The other thing that I want to mention here, which is very, very important, is the fact that it is not one-to-one. Okay, or actually, before I just move off substance P, I want to say one more thing about substance P, and then I'll come back to what I was about to say. Okay, the other important thing to say about substance P is that there is no efficient way to remove it once it has been released. Okay, so often what happens is it spills over onto other neurons besides just the one that it was released onto. So they'll spill over onto the dendrites of other neurons, and it can therefore activate much more than just this one neuron that you actually synapse with. Okay, so that's an important point that's different for substance P compared to glutamate. Okay, right. So, the thing that I was about to say is the fact that it is not one-to-one. It's not one primary nociceptive afferent coming in and synapsing onto one spinothalamic projection neuron. Instead, loads of primary nociceptive afferents from loads of different neurons all synapse onto the same spinothalamic projection neuron. So these spinothalamic projection neurons have loads of different nociceptors coming in and synapsing on them. Okay, and this is one of the things that gives rise to the phenomenon of referred pain. Okay, which is when pain from a visceral organ is felt as pain from a peripheral area. Okay, now the classical example of this is when someone is having a heart attack, a myocardial infarction, you get pain in the left shoulder. Okay, now let me explain why this occurs. So basically, you will also be getting nociceptors from the visceral organs coming in and synapsing on these same spinothalamic projection neurons. So let's say this is one of these silent nociceptors from a visceral organ here coming in and synapsing onto this same spinothalamic projection neuron here. Now, these silent nociceptive neurons are usually not active, okay? It's very rare for them to be active, okay? So if inflammation has occurred, which indeed it has in a myocardial infarction, okay, if inflammation has occurred, however, these can become active and they can start activating the spinothalamic projection neurons and then causing pain. However, when the brain receives this signal, it doesn't know which actual primary nociceptive afferents have actually activated this spinothalamic projection neuron. Okay, so what it does is it assumes that it was the primary nociceptive afferents from peripheral locations that activated it, because that's the more usual situation, basically. It's very rare for these primary nociceptive afferents from silent nociceptors to actually be active. Okay, so it's unlikely that it's come from the peripheral nociceptors, basically. And therefore, it attributes the pain to whichever peripheral location these primary nociceptive afferents innervated. And in the case of the heart, it seems as though the same spinothalamic projection neurons carry information to the brain for nociceptors innervating the left shoulder as for the heart. And that's why the pain from the heart in myocardial infarction is referred to the left shoulder. The brain interprets it as pain in the left shoulder because that's a more usual place for pain signals to be coming from than the heart. Okay, right. So we'll call it there for this video, and in the next video what we will do is we'll move on to where these spinothalamic projection neurons are actually going to go to. So we'll look at how they ascend up the spinal cord, where their axons are going to go firstly. We'll look at how they cross to the other side, how they ascend up the spinal cord in the spinothalamic tract, then where they go to in the thalamus, and then where from the thalamus the signal is then going to go. Okay, so that's in the next video.